Alright, just assalamualaikum everybody. How are you all? I hope that you all are doing well. Welcome or welcome back to my YouTube channel. So this is the part 2 video where we will be solving the yearly past paper of AS level biology 9700. The variant is 22. We are going to solve paper 22 of the year May-June 2025. We are already done with question 1 till question 3 in the previous video, and now we are going to be continuing with question number 4, 5 and 6 in today's video. So let's start the video. Alright, so now let's move on to question number 4 over here. Question number 4 over here says that the tuberculosis is an infectious disease that is caused by the bacterial pathogen. The pathogen has mechanisms to avoid digestion by the phagocytes. Macrophages may engulf the bacteria, but in some cases the bacteria remain alive within the cells instead of being killed. Part A says that although there are two main species of the bacterium causing TB, we know that there are two species or two pathogens that may cause tuberculosis. One is the Mycobacterium tuberculosis, and the other one is the Mycobacterium bovis, right? Now the Mycobacterium tuberculosis is usually found in the lungs, and the Mycobacterium bovis is found in the intestine, right? It is rare for the species that causes bovine TB in the cattle to infect humans. Now they are basically talking about the Mycobacterium bovis. So they are saying that Mycobacterium bovis is a rare one, right? And then they are asking us in the question that name the species of the bacterium that is the main cause of TB in the humans, right? So the main cause of the TB in the humans is the Mycobacterium tuberculosis. So we will write our answer in a way that Mycobacterium tuberculosis. And we rarely have, I think, 3 to 4 infectious diseases in the syllabus. So make sure you learn the spelling of each and every pathogen causing the specific diseases correctly, because in the marking scheme it specifically mentioned that the spelling must be correct. So then only you are going to get 1 marks. Now in part B over here they are saying that explain how the pathogen named in A is transmitted from a person with a disease to a person who is non-infected. So basically Mycobacterium tuberculosis is transmitted by the airborne respiratory droplets, right? So it is an airborne disease which is transmitted by the airborne respiratory droplets of a person who is infected. So how can we jot down its answer? Basically we can jot down its answer in a way that TB is transmitted by the aerosol droplets, or you can say the droplets of the infected person, right? For example, when an infected person will cough, sneeze or talks to another person, and the water droplets, the airborne droplets comes out of his mouth while talking or while he sneezes, coughs, and the uninfected person when inhales those airborne droplets containing the Mycobacterium tuberculosis. So that is how basically the pathogen will be transmitted from the infected person to the uninfected person basically, right? So this is how you are going to answer this. Now the part C over here is saying that state the type of the therapeutic drug which is used to treat the TB. So it is basically antibiotics, because antibiotics are very efficient for the bacterial infections, and as we know that TB is caused by a pathogen which is bacteria in nature, and the antibiotic can kill bacteria very much efficiently. Now what is the catch over here is this, that you should remember for your knowledge that during the treatment of the TB, the 3 antibiotics are given in combination. 3 antibiotics are given in combinations. Now the question over here arises is that why we are giving 3 antibiotics in the combination? Because we give 3 antibiotics because there is a very little chance for the bacteria to develop resistance to all of those 3 antibiotics, right? So for example, because we know that the bacteria develop the resistance towards the antibiotics. So if bacteria develops the resistance against one antibiotic, for example, right, so the remaining 2 antibiotics are going to kill the bacteria. So it will ensure that all the bacteria are killed. Alright, so now let's move on to part D of the question. Part D over here says that figure 4.1 is a photomicrograph of a section of a lung tissue taken from a person who has not been infected with the bacterial pathogen and who does not have TB. So basically this is the figure which they are talking about, and I have also added the reference images from the Google for your reference. Now the question further says that blood vessels and some structures of the gas exchange system are visible in figure 4.1. On figure 4.1, use a label line and label a bronchus, bronchioles and the blood vessels. So let's start with the easy one, the blood vessels. So the blood vessels in the photomicrograph are going to look something like this. We know that the vein has a wider lumen than the arteries, and arteries are having a narrow lumen as compared to the veins. And the arteries are having a thicker wall compared to the veins, which are having a thinner wall. So if I label the arteries or veins and the blood vessels on the photomicrograph, so these are going to be the blood vessels. These will be the blood vessels in the photomicrograph. And then if I talk about the bronchiole over here, so if you refer to this diagram over here, so this is where the bronchiole is starting off. First comes the trachea, the bronchus, and then there comes the bronchioles. And now this image over here, if you see, this is basically starting off with the bronchiole. So we can label this structure as the bronchiole over here. And then this bronchiole is basically divided into the terminal bronchiole. Then there will be the respiratory bronchiole, and then the alveolar duct will come, and then the alveolus will come. So these structures which you are seeing over here are basically the alveolus. These structures over here are the alveolus. So if in case they ask you to mark the alveolus in the further examination question, so you can mark these structures as the alveolus over here. And this is the bronchiole, I already told you guys. Now if you talk about the bronchus, so bronchus is comparatively wider than the bronchiole. So as we have already discussed the bronchiole over here, so most probably this is going to be the bronchus over here, right? So this is how you are going to mark and identify the bronchus, bronchiole and the blood vessels in the photomicrograph. I hope that this is clear to all of you. Alright, so now let's move on to part E over here. Part E over here basically says that in some people with TB, areas known as granulomas may form in the lung tissue as a part of an immune response to the pathogen. So, further in the question, they are saying that figure 4.2 is a photomicrograph of a granuloma in the lung tissue. So this is the figure 4.2, and basically they know that at this level you may not know about the granuloma. So basically they have told us that what basically the granulomas are. So what basically are the granuloma? So they have given us in the question that the granuloma are basically the multinucleated giant cells which are formed by the fusion of the many macrophages. So basically what happens is that when the immune system tries to isolate the mycobacterium tuberculosis which is inside the lung tissue, then the granulomas are formed. So it basically consists of the dead cells and the infected cells, which are mainly the macrophages. So this is the darkly staining white blood cells in the outer areas of the granuloma. This is the darkly staining white blood cells in the outer area of the granuloma. Now further in the question they are saying that with reference to figure 4.1 and figure 4.2, describe and explain how the changes that occur are the result of the granuloma formation. So basically now they are saying that how the granuloma formation can affect the gas exchange and harm the health of the infected person, and also help to prevent the TB developing in the other parts of the body. So basically we are going to answer it one by one. It is of 5 marks, so we can give some points for this statement and then we can give some points for this statement. But we can answer over here that how the granuloma formation can affect the gas exchange and harm the health of an infected person. We know because there is the accumulation of the dead cells over here, right? This is the lung tissue, right? And the alveoli over here, there is the accumulation of the dead cells, right? So if there is the accumulation of the dead cells, the immune cells obviously, so it will make the gas exchange inefficient. It will make the gas exchange inefficient, right? And because the granulomas are taking up the space in the lungs, so they are basically destroying the capillaries, they are destroying the alveoli. So what will happen that because of this, the surface area for the diffusion of oxygen will also be reduced, blocking the gas exchange, right? So also what can happen that the blockage or scarring around the airways because of the you can say the dead cells and the infected cells, what may happen that the airflow may be reduced, so the less air may reach the alveoli, right? So we can answer this question in a way that the granulomas in the lung tissue, what do they do basically? Because the granulomas are forming the clusters of the dead cells and the infected cells around the basically the lung tissue. So what are they basically doing is that they are reducing the number of the functioning alveoli and the capillaries, right? And if the alveoli and the capillaries are reduced, because they are the main region for the gas exchange, so what will happen that the surface area for the gas exchange is also decreased. Dead cells within the granuloma hinder the diffusion of the oxygen and restrict the air movement as well. As a result, less oxygen enters the blood, causing fatigue, breathlessness, and the poor health, right? And then the next part over here basically is saying, asking us to tell that how the granuloma formation may help to prevent the TB developing in the other parts of the body, because obviously it is preventing the TB by trapping the infected cells, right? Because the cells which are infected by the mycobacterium tuberculosis, the immune system is destroying those cells, and that is why the granulomas are forming, right? So by trapping the infected cells and the bacteria inside those cells, the granulomas localize the infection, because we know we can clearly see in the photomicrograph where the granulomas are basically found. So the granulomas tend to localize the infection so that the bacteria cannot easily enter the bloodstream, because it is surrounding infected cell by the macrophages, lymphocyte, multinucleated giant cells over here, right? So it acts as a barrier. So how can we answer this question that the granulomas helps to localize the infection as bacteria and infected macrophages are trapped inside surrounding layers of the white blood cell. For example, the macrophages, they prevent the pathogen from spreading into the circulation. The immune response, including the antigen presentation, cytokine release, because we know that when the white blood cell go and it fuses with the, or it responds to the infected body cells, so it can do the immune response in various ways. For example, antigen presentation, cytokine release, right, phagocytosis. So it helps to stop the mycobacterium tuberculosis from multiplying and spreading to the other part of the body, right? So I hope that this is clear to all of you. Alright, so now let's move on to question number 5 over here. Question number 5 over here says that there is a global shortage of blood for the transfusions. Researchers can culture bone marrow stem cells in the laboratory to manufacture red blood cells for the potential use as an artificial blood product. The researchers collect the bone marrow stem cells that are present in the small quantities in the blood rather than extracting them from the bone marrow. So what are basically the stem cells? Stem cells are the undifferentiated cells of the body that can differentiate into specialized cells, right? So they are saying that researchers have collected the bone marrow stem cells that are present in small quantities in the blood rather than extracting them from the bone marrow themselves. So they have extracted the bone marrow stem cells from the blood, and they are saying that because there is a global shortage of the blood transfusion, so the researchers are culturing the bone marrow stem cells in the laboratory to manufacture the red blood cells. Now further in the question they have said that the antibodies specific to the bone marrow stem cells are attached to the tiny magnetic beads. The beads are then added to the sample of the blood. An electric field is applied to immobilize the beads so that the bone marrow stem cells can be collected. Now in part A they are saying that the antibodies are attached to the beads so that the sites used for the binding to specific molecules on the bone marrow stem cells are left exposed. Name the term given to the specific molecules on the bone marrow stem cells that attach to the antibody binding sites. So basically if I talk about the structure of the antibody over here, For example, if this is the structure of the antibody, so these are the antigen binding sites in the antibody. So obviously, for example, if this is a bone marrow stem cell and these are the antigens on the bone marrow stem cell, so these antigens are going to bind with the antibody at the antigen binding sites. So they are saying that name the term given to the specific molecules on the bone marrow stem cells that attach to the antibody binding sites. So the antibody binding sites are basically the antigen binding sites. So the antigen of the bone marrow stem cell is basically going to attach to the antibody binding site. So the answer would be the antigen, right? Now let's move on to part B over here. Now the part B over here says that suggest and explain the advantages of using the bone marrow stem cells from the blood sample to manufacture the artificial red blood cells. Now basically the bone marrow stem cells are naturally able to produce and differentiate into the red blood cells as well as the other blood cell types if needed. So basically if they are able to differentiate into the red blood cells, so this ensures that the cells that are basically produced are efficiently produced, right, because it is producing from the stem cells which we have extracted from the body. So the desired red blood cell is being produced. This is an advantage of it. So we can say that the bone marrow stem cells can differentiate into the red blood cells. So we can say that the bone marrow stem cells can differentiate into the red blood cells. Also because the stem cells have the ability that they can divide continuously and they can produce a large number of cells. So we can say that this will allow the mass production of the red blood cell. This will allow the mass production of the red blood cell from a very small population. You can say that with a relatively small starting population, like we will start with a small population of bone marrow stem cells and they will divide continuously and continuously producing the large number of genetically identical red blood cells. So we can say that these two are the advantages of the manufacturing cells using the bone marrow stem cells. Then part C, they are saying that now after being done with part B, let's now move on to part C over here. Part C says that one desirable feature of the artificial blood products like the blood cells which are produced artificially, such as the artificial red blood cells, is that they should be economical to produce, like they should not be heavy on the pockets and they should be cheap, right? Suggest other desirable features of the artificial blood products. So the other desirable features of the artificial blood products which are made in the laboratory should be that they should not have any toxic effects on the body. Like for example, if we are producing the white blood cells artificially in the lab, so when we are inserting those white blood cells into the human body, then there should not be any toxic effects of those cells in the human body, and they should not stimulate any immune response in the recipient. Also, it should not carry any pathogen that may cause harm in the human body. So these are some of the features of the artificial blood products that should be taken into consideration before we inject those artificial blood products into the human body. Alright, so now let's move on to question number 6, all of you. By the way, in the previous question, if my voice was low, so it was because I was sitting in the library of my university. So I was recording in the library with a mic. So that's why my voice is a little lower there. I hope that it is okay for you guys. Now let's move on to question number 6, all of you. Now question number 6 over here says that horseradish peroxidase, which is HRP, is an enzyme that can be extracted from the roots of the horseradish plant Armoracia rusticana. HRP is used extensively in the industry and the technology. In the reaction catalyzed by the HRP, hydrogen peroxide is used to oxidize an organic substrate. This is summarized in figure 6.1. So in this they have told us that an organic substrate is combined with the hydrogen peroxide in the presence of the HRP, which is Horseradish peroxidase enzyme, and then the oxidized substrate and the water is released, right? Then in part A over here they are saying that inhibitors can have an effect on the Vmax, the maximum rate of the reaction. Vmax is the maximum rate of the reaction of the enzyme, right? And the Km, the Mitchell's-Menten constant of the HRP, which is the enzyme. So what is the Mitchell's-Menten constant? Basically it is the substrate concentration at half Vmax, right? What is the Mitchell's-Menten constant? It is the substrate concentration at half Vmax. So if they have asked you the Km over here, then it is a high possibility that in the October-November papers of October-November 2025, they may ask you to write down the full form of the Km, or they may ask you that what is Km. They can ask you that define Km, right? And also they can ask you the effect of Km on the competitive and the non-competitive inhibitor, right? So you should be ready for all of these questions. Now they are saying that figure 6.2 shows the effect of the substrate concentration of the rate of reaction of HRP. Now complete figure 6.2 by drawing a curve to show how the presence of a non-competitive inhibitor. Now a key term over here, or a key concept over here, is the non-competitive inhibitor will affect the rate of the reaction of the HRP. So we know that there are two types of the inhibitors. One is the competitive inhibitor and the other one is the non-competitive inhibitor. Now what we have to do is we have to use the curve you have drawn to obtain an estimate of the Km. Alright, so first what we have to do is that we have to draw the curve in the presence of the non-competitive inhibitor, and then we have to estimate the value for the Km. So in the competitive inhibitor, what is happening, that the Vmax is same but the Km is different. Vmax is same but the Km is different. But in the non-competitive inhibitor, what is happening, that the Km is same but the Vmax is different. Km will be same but the Vmax will be different. So as the Km is same, so can we identify the Km over here, that what would be the Km of this value of this graph? Can we identify the Km? Km is the substrate concentration at half Vmax. So half Vmax over here is 8, right? So half Vmax would be 8 divided by 2, which will be 2, 4, 6, 8, 4, right? So at 4, the substrate concentration over here is 0.01, 0.01, 0.02, 0.03, 0.04, 0.05, 0.06. So the substrate concentration over here is 0.06, right? And then now they have asked us to draw the curve as well. So how we are going to draw the curve? We are going to show a different, you can say we are going to show a different Vmax over here, which is basically below the original graph. This is the original graph, right? And the non-competitive will be below. So we will draw it like this, right? We drew it like this, and then this is the Vmax over here for the non-competitive inhibitor, right? So this is the graph that we have drawn. Now use the curve you have drawn to obtain an estimate of the Km. You can obtain the estimate of Km. For example, Vmax of the non-competitive inhibitor is 4, and then when we do it, 4 divided by 2 is equal to 2, right? So on 2, the substrate concentration is 0.06. As the Km is basically unchanged, so you can use the first graph to determine the Km as well. Or you can draw the other graph and then you can determine the Km. It's up to you, but that's why they have written the estimate, right? So The correct answer over here is 0.06. Now let's move on to part B over here. Part B over here basically says that the scientists can design the synthetic DNA nucleotide sequences to produce a synthetic RHP gene. These sequences will include a start codon and a stop codon. So we know basically the start codon and the stop codon is made up of the three nucleotides, so that the translation of the messenger RNA, the mRNA, can occur. We know that what is translation. So translation is a process where the mRNA basically forms the polypeptide chain or the protein chain, right? And then further in the question they are saying that explain what is meant by the start codon and a stop codon. So this is a very easy question and a mark gaining question as well. So the start codon and the stop codon both are found on the mRNA, right? Now, what is the definition of the start codon? It is the triplet of the nucleotides in the mRNA that signals for the beginning of the translation. And then what is the stop codon? Stop codon is a triplet of the nucleotides in the mRNA that signals for the end of the translation. So this is what is meant by the start codon and the stop codon. Now part C over here says that the HRP, which was the enzyme, right? HRP, which was the enzyme, is used in an immunological test known as the sandwich ELISA. So what is ELISA? ELISA is a biochemical technique which is used to detect the antigens or the antibodies in the sample. Detect antigens or antibodies in the sample. And what is the full form of the ELISA? For your knowledge, I am telling you that ELISA की full form होती है enzyme linked immunosorbent assay. So it is a biochemical technique which will detect the presence of the antigen and the antibodies in the sample. One use of the test is to diagnose the disease. Now figure 6.3 outlines the main steps in the sandwich ELISA in which a toxin released by a pathogen is detected in a sample of the body fluid taken from a person who is ill. The test involves two types of monoclonal antibodies that can bind to the toxin, a capture antibody and the detection antibody. Now, as the name suggests that it is a sandwich ELISA, so what will happen that, for example, if I draw an antibody over here and this is for suppose the foreign antigen or you can say the toxin, so what will happen that the another antibody will come and bind to it, right? And the antigen is basically sandwiched between the two antibodies. That's why it is known as the sandwich ELISA, right? Because the antigen, which is a foreign particle, and foreign particle can be the toxin as well. So toxin is treated over here as the antigen or the foreign particle which is basically sandwiched between the ELISA, a sandwich between the antibodies. ELISA is the whole technique. Now they have given us the 6 steps over here. Let's understand them. Step 1 is that prepare a test well by immobilizing the captured monoclonal antibodies to the support surface. So the captured antibodies are resting on the basement surface of the test well. Then step 2 is remove the sample. Remove a sample of a body fluid from a person who is ill and add the sample to the test well. So as the body fluid is from the person who is ill, so it will have the toxins as well. So the toxins, whatever the toxins will be there in that body fluid of a person, so what will happen that the toxins will come and it will bind to the binding sites on the antibodies. And toxins, we know that the toxins are the antigens over here, which is antigens or you can say the foreign bodies over here, which will stimulate the immune response. Then step 3 will be that wash out the test well to remove any substance not bound to the capture antibodies. Now we will give time for the capture antibodies to bind with the toxins, right? Toxins from the fluid of the toxins from the body fluid of the ill person. Now the toxin came and then they bind to the monoclonal antibodies which were resting on the basement surface of the test well, and then we will wash the test well to remove any substances not bound to the captured antibody. And then in the step 4 what we are going to do is that we will add the detection antibody to the test. Now what is the detection antibody? The second antibody that we will add to the test well is known as the second antibody that we will add to the test well is known as the detection antibody. Now this detection antibody, if you guys notice, so this detection antibody is linked with the HRP enzyme over here as well. So whatever detection antibody we are having, these antibodies are linked with an enzyme, right? And then what they will do, that they will come and they will bind to the they will come and they will then bind to the toxin. The detection antibody will come and it will bind to the toxin. Over here, this is the enzyme HRP which I am showing, right? It will come, it will bind to the toxin, and then step 5, wash out the test well to remove any detection antibody that is not bound to the toxin. Alright, now the step 6 is that add the substrate for catalysis of the HRP and add acid after a set time to stop the reaction. Then what will happen, that when the detection antibody will come, it will bind to the toxin. So what will happen, that the substrate is added, right? The substrate for the HRP enzyme, the substrate for the HRP enzyme is basically substrate for the HRP enzyme is added, and then this HRP enzyme is basically going to act on this substrate, and when this HRP enzyme is going to act on the substrate, so it will produce a color change, right? So it will produce a color change. So by this color change we can identify the antigens. Then in part 1 they are saying that complete the figure 6.4 to show the situation in the test well after the step 5. So after step 5, what was the step 5? Basically wash out these test well to remove any detection antibody that is not bound to the toxin. So after step 5 they are saying, and before step 6, so what will happen, that the detection antibody will bind to the detection antibody, we will show that the detection antibody is bound to the toxins on the capture antibodies. This is the capture antibody, this is the toxin, this is the detection antibody. So we will show that the detection antibody is bound to the toxin which is on the capturing antibody, right? So this is how we are going to complete the diagram. We can also show one more detection antibody attached to the toxin of the capture antibody. So this is how basically we are going to draw the this is basically how we are going to draw and complete figure 6.4. I hope that this is clear to all of you. Now let's move on to part 2 over here. Part 2 over here says that with reference to figure 6.3, describe one difference between the capture antibody and the detection antibody. So we know that the capture antibody is immobilized, it cannot move, right? It was stuck on the well surface throughout the whole process, right? So we will say that the capture antibody is immobilized on the well surface, whereas we know that the detection antibody binds to a different side of the toxin and has the HRP enzymes attached to it as well, right? You can say that the capture antibody is immobilized, it is not mobilized, whereas the detection antibody is mobilized, it can move. And also the detection antibody has a antibody binds to a different side on the toxin, right? For example, the capture antibody was binding to this side of the toxin, so the detection antibody is binding to a different side of the toxin, you can say, right? And then you can say that the detection antibody has the enzyme attached to it, whereas the capture antibody had no enzyme attached to it. So this is how you are going to answer the part 2 over here. Now let's move on to part 3 over here, and part 3 finally the last part of this paper and this question as well, and it is of 4 marks. Also I can see over here that we have to use our knowledge related to the colorimeter, and there are the suggestions given over here as well. So let's read part 3 first of all. Part 3 over here is saying that figure 6.5 outlines the reaction catalyzed by HRP in step 6. The organic substrate TMB is in excess and changes the color when it is oxidized. As I told you that whenever the HRP is going to as I told you that whenever the HRP is going to whenever the enzyme that is as I told you that whenever the enzyme that is attached to the detection antibody, and in this case it was the HRP, whenever this enzyme will be given its substrate, which was the organic substrate, TMB is the organic substrate, hydrogen peroxide, it will react, right? The HRP enzyme will react with the substrate and it will produce a color change, right? It will produce a color change, and the color change will be proportional to the concentration of the antigens. So this indicates a positive result, and a color change is basically produced in which the oxidized TMB becomes blue green and the water is released as a byproduct. Now a student suggests that a low concentration of toxin may be diagnosed as a negative result instead of a positive result. And then another suggestion is that using a colorimeter after step 6 in the sandwich ELISA test would provide a quantitative result and help to avoid this error. With reference to figure 6.5, explain why using a colorimeter would provide a quantitative measurement for the detection of low concentration of toxin and help to confirm a positive result. So toxin over here, as I told you that the toxin over here is the antigen, right? A foreign particle which is producing an immune response. Now let's try to understand this question. अच्छा, यहाँ पे जो एक चीज़ आप लोगों को समझने की ज़रूरत है, द थिंग यू वांट टू अंडरस्टैंड ओवर हियर फर्स्ट ऑफ ऑल इज़ दैट द कलर चेंज, वट एवर द कलर चेंज विल बी हैपनिंग विल बी डायरेक्टली प्रोपोर्शनल टू द concentration of the antigen, right? It will be directly proportional to the concentration of the antigen. Now how it will be directly proportional to the concentration of the antigen? Now we know that more the toxins will be there in the sample, more toxins, if there will be more toxins in the sample, then more toxins will go and bind to the capture antibodies, right? And if more toxins will go and bind to the capture antibodies, then there will be more detection antibodies which will bind to those toxins, right? And if there will be more detection antibodies with the HRP enzymes, we know that the HRP enzyme was attached to the detection antibody, right? And if there will be more detection antibodies attached to the toxins, then it means that automatically there will be more HRP labeled detection antibodies, right? Or you can say that there will be more detection antibodies with the HRP enzymes, and as more HRP enzymes will be there at the detection antibody, right? So what will happen, that it will catalyze more TMB. It will catalyze more TMB, and if you remember that TMB was a colorless solution which was getting oxidized and it was changing its color to blue green. So as there will be more TMB, so there will be more reaction, right? More HRP, so there will be more TMB that will be oxidized, and if more TMB will be oxidized, so it will mean that there will be a stronger color intensity. So we can clearly say that the color change over here in the solution will be directly proportional to the concentration of the antigen, or in this case you can say the toxins as well. So therefore we can say over here that the color intensity is directly proportional to the toxin concentration. Now how do we have to answer this? Question? So we can answer this question in a way that we can tell about the concentration of the toxins. We can say that a low concentration of toxin may produce the less HRP detection antibody complexes, and due to this, what is happening, that the less oxidizing of the TMB will take place, and if there will be less oxidizing of the TMB, then it will give a paler color which may not be visible to the naked eye. So a colorimeter, what does a colorimeter will do, how it can change the result, that it will measure the absorbance of light by the reaction mixture, and it will give a quantitative reading. So this allows a small difference in the
detected
color
as a
density
positive
to
result
be rather than the whole negative result. So then we can use the calibration curve with the known toxin concentrations to allow the exact concentration of the toxin to be determined. So we can answer it in a way that colorimeter can measure. Colorimeter measures the absorbance of light by reaction mixture, and we know that a low concentration of the toxin produces a pale color, giving a low absorbance reading. At least it is giving a low absorbance reading. It is not giving a negative reading, right? If there is a color change and if there is a less color change, so it is not
it
being
will be counted as a positive
result, but with a low absorbance reading. So the different absorbance reading distinguish a low positive result from a negative result. Due to this, you will avoid your reliance on the visual judgment as well, which can involve the human error. So due to this, what will happen that because the different absorbance reading is being readed on the colorimeter, it will reduce your reliance on the visual judgment, which can contain the human error as well. So we can say that it can avoid the reliance on visual judgment, right? So then you can say that using a calibration curve with the standards allow the toxin concentration to be determined quantitatively. So this is basically how you can answer this question. I hope that this is clear to all of you. Alright, so that was it for this video. We have completed the May-June 2025 paper 2 yearly past paper of AS level biology. I am also attaching the link of the part 1 of this yearly past paper where I have solved question 1 to question 3 in the description below. You guys can go to that link and check that out as well. Also, I am starting a yearly past paper session of paper 2, and in case any one of you is interested to enroll in it, you guys can contact on the following number, right? So you guys can contact on this number that you guys are interested to enroll in the paper 2 yearly past paper session. So our team is going to contact you. So that was it for this video. I hope that you guys understood each and everything. If you guys have any confusion, you can also message me or you can comment down in the comment section below and I'll try to answer your queries, right? So if you benefit from this video, do remember me in your prayers. Take care. Allah Hafiz everyone. See you guys in another past paper solution.